All right, welcome everybody. This is the October 24th edition of Micro Seminar. I'm really happy to introduce Steffi Lutz today. She is doing her second year, well, she's been postdocing for 2 years at the Helmholtz Center in Potsdam. Before that, she did her PhD at University of Leeds and a combined undergraduate and master's degree at the University of Tübingen in Germany as well. And so today she's going to be talking to us about glacial microbiology. And without any further ado, one more ado actually, as usual, if you have questions, please tweet them to the hashtag #USeminar or @microseminar on Twitter, and I'll make sure to get those questions into Steffi as we go. Okay, now without further ado, Steffi, go ahead and take it away.
Okay, thanks very much. I'll just share the presentation. Where is it? All right. Can you see it?
Right now, it's just showing the Hangout screen.
Okay. That's odd. Okay, sorry, I'm just trying to get this started.
That's all right. How you guys doing?
So far, everything's going well.
Okay. So driving kids to school, so I just want to say hi and hello and bye, Biddle.
Can you see the presentation now?
Yeah. Yes, for the most part, and just try to go into presenter mode now.
Okay, it was on my screen.
Yes, for the most part, and just try to go into presenter mode now.
Okay. Now it was on my screen. Can you see the whole presentation?
It looks the same as before.
Okay. And now?
It's still the same.
Okay, I'll send them out on my screen.
There you go.
Yeah?
Perfect.
Okay, perfect. Okay, so finally we can get started. Sorry about that. Okay, so hello everyone. Good morning or good evening, wherever you are. Thanks for tuning in. It's a great pleasure for me to present in the seminar series, and I would like to thank the organizers for giving me this opportunity. I'm a postdoc in Liane Benning's group at the German Research Center for Geosciences, and I would like to present some of our work on glacial microbes. I would like to begin with just a few facts why glaciers and ice sheets are important, which you're probably all aware of. They're prominent features on our planet and currently comprise about 10% of the Earth's surface. They have been prominent throughout Earth history, with a highly varying arid extent of the ice cover during glaciation and deglaciation phases. For example, during the last glacial maximum, which was about 20,000 years ago, we had 32% ice coverage, and during the so-called snowball Earth phase, which was about 650 million years ago, near complete glaciation was reached. Then the ice field plays a critical role in the global hydrological cycle, since approximately 70% of Earth's freshwater is stored as ice, and therefore it is tightly coupled to global climate change and sea level rise. Furthermore, glacial microbiology is a very young research field, and only recently glaciers and ice sheets have been recognized as one of Earth's biomes. A plethora of organisms can be found on glacial surfaces. That includes bacteria, archaea, algae, fungi, and even insects, tardigrades, nematodes, and other small metazoans. But these ecosystems are exclusively driven by microbes, which makes them very unique. Supraglacial ecosystems comprise two big ice sheets, Greenland and Antarctica, and polar and alpine mountain glaciers, circled here in red, for example, the Alps in Europe, or glaciers in Svalbard in Iceland. They differ considerably in terms of their area extent, and the two major factors for microbial growth, the availability of liquid water and nutrients. All polar and alpine mountain glaciers together are by far much smaller than the two ice sheets. However, on most mountain glaciers, there's enough liquid water available during the melt season to drive microbial growth. And because of their small size, they're even more strongly connected to adjacent ecosystems, such as glacial forefields. And therefore, they are likely more seeded by material blown onto the ice surface, such as allochthonous carbon. In contrast, the Greenland and Antarctic ice sheets experience much lower nutrient loadings, in particular in the interiors. The Greenland ice sheet has experienced extreme melt events in the last years, which can result in up to 100% of the surface being affected by surface melting. And these events will likely reoccur more frequently in the future. And this makes the Greenland ice sheet the largest supraglacial ecosystem. Nevertheless, and despite the geographic differences, the glacial habitats that can be found worldwide are remarkably similar in terms of the main primary producers and ecosystem engineers, which I will introduce in the next slides. The main primary producers in snow are snow algae. They are unicellular freshwater algae that form extensive blooms after the onset of melting. Melting snow can turn into shades of green, red, and yellow due to snow algal pigmentation. Green snow is generally caused by young flagellated chlorophyll-rich snow algae. And with increasing light irradiation, both visible and UV, they produce carotenoids to prevent photoinhibition and causing a reddish coloration of the snow. And this is accompanied by the formation of spores, which are still photosynthetically active. But in winter, when the cells get buried by snow, they become dormant until the next melt season. Snow algae are widespread and have been described in numerous places in the Arctic, Antarctica, and alpine settings. Red snow is also sometimes called watermelon snow and has already been observed by the ancient Greek Aristotle. But then only in the 19th century, with the development of microscopy, the red snow phenomenon could be linked to algal blooms. And you might have already heard of the famous Chlamydomonas nivalis, which has traditionally been associated with red snow. However, many species have been wrongly classified as Chlamydomonas nivalis, also because species identification by microscopy is very challenging, since the cells undergo dramatic morphologic changes throughout the melt season, and then only genetic analysis could reveal that there are actually several species causing red snow. And Chlamydomonas and also Chloromonas nivalis therefore have to be regarded as collective names. They're not single species. Green snow is usually less common since it's often very short-lived transient stage. And the carotenoid-rich red stage is much more widespread and more long-lived in the season. However, not all green snow turns red, and the triggers and mechanisms of this transition are still not understood. And finally, you've probably all heard of yellow snow, but not that the one that is also caused by algae. These are completely different algae, and they are much less often described. Then when the snow cover disappears, bare ice surfaces are revealed, and melting clean ice can turn into so-called dirty ice or gray ice. The gray coloration is also caused by pigmentation of algae, ice algae, and these ice algae had already been discovered by Arctic explorers in Greenland in the 1870s. However, only recently they've been rediscovered, and so only like in the last 10 years. And it has been recognized that the grayish coloration of the dirty ice habitat is not just inorganic dirt, so to speak, but also due to their brownish pigment, which was identified by Remias et al. As a hydrophilic tannin-like pigment. On the bare ice surface, there are also so-called cryoconnite holes, which form on darkened ice due to the accumulation of particles and the preferential warming and melting. And the primary producers in these are bacteria. And in the Arctic, the cryoconnite holes are usually filled with liquid water and cover between 1 to 10% of glacial surfaces. And in Antarctica, they are often completely frozen or have an ice lid on top. Prominent algae in ice algal blooms are Ancylonema nordenskiöldi and Mesotanium berggreni. Mesotanium can be found in polar and alpine regions, whereas Ancylonema so far has only been found in the polar regions. And we know by far less about ice algae compared to snow algae. It's probably because they've been overseen for such a long time due to their grayish coloration, which makes them basically look like dirt, and which is much less striking than the green and red coloration of snow algal blooms. All glacial microbes, including snow and ice algae, must overcome a multitude of environmental stress factors, and they have evolved a range of adaptations to counteract the damaging effects of low temperature environments. These include freezing of water, desiccation, nutrient deficiencies, high light irradiation, visible as well as UV, which is especially high between the snow and ice crystals, where light is even more reflected. And they're constantly exposed to freeze-thaw cycles, and therefore experience a lot of osmotic stress. And this also controls the availability of nutrients, whether the nutrients are dissolved and available or not. And to give you an impression of these freeze-thaw cycles and how often they alternate, here's a short time-lapse video we took on a glacier in Greenland over a few days. And as you can see, it changes very quickly between freezing and thawing, which causes a lot of osmotic stress for the algae. So the cryophylic algae are polyextremophiles, and this makes them of general interest to study a wide range of fundamental cellular processes. And here you can see a schematic of the glacial ecosystems. The algal blooms are part of the supraglacial ecosystem, which is by far more sampled and studied than, for example, the englacial and subglacial ecosystems. However, the spatial coverage of the supraglacial ecosystem is also still highly patchy due to the extreme remoteness of many areas. The extent of the snow cover can vary immensely between glaciers and will either partly or completely disappear during the course of a melt season. At the beginning of the melt season, snow algae are the main primary producers, and then later in the melt season, the snow line and the snow algal blooms move higher up, and bare ice is exposed, and ice algae become the main primary producers on the ice. And these changes can happen extremely fast, and we follow them, the changes in habitats from the beginning of the melting season over a 3-week period on a mountain glacier in Greenland called Middelburg Glacier. This was in July 2012 during one of the extreme melt events in Greenland. And in the bottom picture, you can see the bottom part of the glacier. And when we arrived at the beginning of the melt season, there was no visible microbial presence, and the whole surface was still slightly covered by snow. Then after a few days of melting, green snow formed at the bottom, caused by flagellated snow algae. And with ongoing melting, a few days later, some of the green snow turned red, and the snow line experienced an elevation gain of about 200 meters, and red snow could be found higher and higher up. More bare ice got exposed at the bottom, which was then dominated by ice algal blooms, and that all happened like in a 3-week period. And these changes in surface habitats are also accompanied by changes in surface albedo. As you probably know, albedo is the ratio of reflected to incident radiation. And snow and ice without algal blooms have a much higher albedo, meaning more light is reflected back. And due to the algal pigmentation, the albedo is decreased, meaning more heat is retained, which causes more melting of the snow and ice. And on this glacier in Greenland I just talked about, we carried out an albedo survey with over 700 measurements of the different habitats, which showed that snow and ice patches with algal blooms have a considerably lower albedo. And this is another good reason to get a better understanding of these algal communities. In terms of their distribution patterns, their community structures, their metabolism, the interactions with other communities, and their contribution to glacial melting. And these are basically the questions that drive the research in our group. And we're working on snow and ice algae, but the ice algae work is all very much work in progress. And that's why we'll focus on our findings on snow algae in this presentation. And in order to answer these questions, we're pursuing a multidisciplinary approach using large-scale omics techniques, but always in combination with targeted analyses, such as targeted metabolite analyses, and a good evaluation of the physicochemical boundary conditions. You can see here that the metatranscriptomics is also in brackets because this is also work in progress and will not be covered in this presentation. I will start with our findings on based on the Amlicon data, which I will be presenting in the next few slides. And in order to answer the question, what are the snow algal community structures and what parameters are controlling them, we conducted a large-scale biogeography study of red snow. If you remember what I said in the beginning, red snow is the most widespread snow algal habitat. And we chose 4 well-separated geographic locations of different latitudes, altitudes, and geochemistry to get a good representation of the Arctic. We sampled 6 glaciers in Svalbard, high up in the Arctic, at low elevations and with higher concentrations of easily leachable elements. Then 6 glaciers in northern Sweden, low latitude but high elevation, and with much higher carbon concentrations, also closer to proper vegetation, like proper trees. And then 8 glaciers in Iceland with completely different bedrock, namely basalt, with high iron concentrations, and also some input of volcanic ash. And then 1 glacier in Greenland, the same I've already mentioned. So quite a good spread of different boundary conditions. So we used the 18S rRNA gene to get the eukaryotic diversity. Back then we used iron torrent sequencing. Now in the meantime we changed to Illumina. We had 32 samples in total, grouped here into the 4 geographic locations. And what you can see on the right is that only 6 algal species made up more than 99% of the community structure in all sites. And despite the differences in the geochemistry and the geography, the community structures were highly similar. The same you can see on the left in the principal component plot, where samples did not cluster into geographic locations, simply because they're highly similar across all locations. So there was no correlation between community compositions and any of the quantified geochemical parameters. And this suggests that they are independent of location-specific factors, and likely the availability of liquid water is the major factor controlling snow algal distribution. So whereas red snow algal blooms seem to be independent of the local snow chemistry, we got completely opposite results for the bacterial communities. Here displayed for bacterial classes, but this was also valid on lower taxonomic levels. So the samples clustered according to geographic locations and found correlations between community structures and local geochemistry. One example are the sphingobacteria, which positively correlated with carbon species, namely total solid carbon and dissolved organic carbon. So the geochemistry is not affecting the snow algal community structure, but what about the metabolism? One of the metabolites we're interested in are the pigments, since these cause the darkening of the glacial surfaces. And on the left, you can see the pigment composition of all the samples from Svalbard in Sweden. And not surprisingly, secondary carotenoids are the dominant pigments in all samples. The secondary carotenoid composition was mostly made up by astaxanthin, which gives the snow the red coloration. On the right, you can see the secondary carotenoid composition on the y-axis and solid carbon to nitrogen and carbon to phosphorus ratios on the x-axis. And I've also included here the green snow samples just for comparison. The black line indicates the optimal Redfield ratio for carbon, nitrogen, and phosphorus. And what we found was a positive correlation for secondary carotenoid production and low carbon to nitrogen ratios, suggesting that secondary carotenoid production is triggered by nitrogen limitation. And in contrast, we did not find such a correlation for phosphorus. Pigments are often stored in lipid bodies, which yield a chloroplast. And astaxanthin, the main secondary carotenoid, is often esterified with fatty acids. and therefore we also looked at fatty acid compositions, which you can see on the left. And you can see the polyunsaturated fatty acids were most abundant. Again, you can find the carbon to nitrogen and carbon to phosphorus ratios on the x-axis and the polyunsaturated fatty acid content on the y-axis. Here we got similar results as for the pigments. There was a link between low nitrogen content, but not for the phosphorus content. So nitrogen limitation seems to play an important role in the metabolite production of these algae, at least in terms of pigment and fatty acids. Finally, we were also looking at algal cell numbers and biomass, and there was some variation in algal cell counts and the total biomass between samples, but on average, no significant differences across geographic locations. Furthermore, we found a clear link between the algal biomass and the albedo measurements, which you can see at the bottom right. The biomass is on the x-axis and albedo on the y-axis. And you can see that a higher biomass correlates with a decreased albedo. So the previously described similarities in community structures, metabolites, biomass, and albedo values allowed us then to develop a simple model for the impact of snow algae on surface albedo over the course of one melt season. So on average, we have about 100 days of melting per season. And before the onset of melting, glacier surfaces are covered by dry, clean snow with an albedo of about 90%. And on the left, you can see the values we got from our albedo survey in Greenland for melting snow without algae and snow with algae. Then we had two scenarios: in blue, just melting snow without algal blooms, and in red, melting snow with snow algal blooms. And the estimated overall decrease in snow albedo integrated over the 100-day scenario was 30%, which so 13% lower albedo, which will lead to higher melt rates. The biggest unknown at the moment is the percentage of glacier surfaces that are covered by algal blooms. But with global warming, this number will likely increase. Because if you remember what I said before, liquid water is likely the major driver of algal growth. And on the right, you can see a map of Greenland during the extreme melt events in 2012. And in this case, red is not algal blooms, but it's surface melting. And in 2012, nearly 100% of the Greenland ice sheet was affected by surface melting, meaning there's a high potential for algal growth. So basically, we have more melting, leading to more algal growth and a further decrease in albedo, and also an early exposure of dirty ice, which has an even lower albedo. So we get this positive feedback mechanism. And at the moment, this bioalbedo effect and biologically driven melting is not considered in any climate models. And in order to integrate it into models, it is important to quantify all the parameters affecting surface albedo. So not only algal blooms, but also physical parameters such as changes in snow and ice water content and grain sizes, as well as geochemical parameters, including mineral dust and black carbon or soot. And then to quantify the contributions of all these parameters. And this can only be done as part of a large team effort and when bringing together different expertises. And we are part of such a large project called Lichen Plume, which is based in the UK and aims to understand the melting of the Greenland ice sheet. And we are part of the particles work package to characterize and quantify all the different particles, organic and inorganic. And then other work packages look at the microbiology and the albedo. And then finally, all the data will be integrated to incorporate biological darkening into climate models. And then that way we can come up with a revised estimate for future sea level rise. It's a really cool project. And here's the website and the Twitter. So please check out the website. There's also a nice blog on how it is to live on the Greenland ice sheet for a couple of weeks. And yeah, please follow us on Twitter. So we will move on now, and I would like to talk briefly about our metagenomics and metabolomics work, which is mostly work in progress. And I would like to present some of the initial results and also current limitations and perspectives. This is a study we've carried out in Svalbard. And if you remember, I said in the beginning that it's still not understood why some snow patches transition from green to red and others don't. And this seemed to be the ideal place because we found these very, very intense green and red snow patches just next to each other. And that's really not Photoshop. That was really that green and red. So we know the community composition from our Ompicon data and decided to sequence the metagenomes to learn more about the community functions. And in order to know what genes are actually derived from the algae, we tried to map the reads to closely related reference genomes. Well, when you have a look in NCBI, there are only 22 sequenced genomes of green algae, with the famous model organism Chlamydomonas reinhardti being the closest match. And this algae is actually a mesophile that lacks the property of cryophilic algae. And therefore, it's also not surprising that actually less than 1% of the shotgun reads could be mapped to the genome of Chlamydomonas reinhardti. We also used several other approaches, such as binning the reads based on the sequence properties, such as K-mer frequencies, but that was also not very successful to pull out the algal sequences. So there was clearly a need for appropriate reference genomes, and we decided a sensible thing to do would be to take one step back and get reference genomes before we can move on and work more with the metagenomes. And we're working on this in collaboration with Thomas Leyer from the Fraunhofer Institute in Potsdam, who is the head of a large cryophilic algae collection. And we chose 5 strains that are closest to the algae in our samples. Not the same, but close enough, because that's another obstacle. These algae are really difficult to culture in the lab, and not all of them happily grow in lab cultures. So we had to choose whatever was available. And we did some initial Illumina sequencing to get an idea of these genomes. And as you can see, the genome sizes are rather large, which will not make it a quick task to get well-assembled and annotated genomes. And also at that point, when I got those numbers, I also started questioning my choice to work on microalgae and not on something much smaller than bacteria. Anyway, last but not least, I would like to present some of our metabolomics data that we're using to help elucidate the life cycles of these algae. Metabolites are the ultimate response of an organism to the environment. And the great thing is that metabolites are conserved across organisms in contrast to genes. So glucose, for example, is the same in all organisms. So should be possible even without appropriate reference metabolomes. So these are data from the same study of the green and red snowfields in Svalbard. And as you can see on the left, we could identify quite a few metabolites and group them into metabolic pathways for the green and red snow samples on the right. And what we found is that the metabolic profile of green snow was characterized by key metabolites involved in growth and proliferation, such as auxine and the tryptophan degradation pathway, which is a plant hormone which has long been known to enhance growth of green algae such as chlorella. And this was also supported by a high abundance of the carbohydrates, ribose and lactose, which we got from the targeted metabolite analyses. And ribose and lactose are part of a variety of molecules involved in proliferation. So that also showed that it's always good to have some complementary data, not just large-scale omics data, but also some targeted analyses. And in contrast, red snow was characterized by storage and reserve metabolites. For example, sphingolipids were more abundant, and they're believed to inherit a variety of functions, including hardening of cell surfaces to protect cells from the harmful environment. And in addition, we observed an upregulation of the uridine monophosphate biosynthesis pathway, which is involved in the synthesis of ribonucleotides, which can possibly be used as storage compounds for overwintering. And this is, of course, just a snapshot. And we need to study more samples in order to make universal assertions. As you can see on the top right, there's also still a large proportion of unidentified metabolites, and studying appropriate reference metabolomes would also be a sensible thing to do in that case. In particular, since again the mesophile Chlamydomonas reinhardti is the closest reference that can be found in the databases. So with my last slide, I would like to give some overview where we are with biomics techniques in general for microalgae in glacial environments. And you've probably all seen this graph before, the next-gen sequencing hype cycle. And I think in this case, it summarizes it quite well. Just the whole cycle is delayed by about 10 years compared to more studied and less exotic environments. It is quite easy to generate a lot of data, but without the appropriate reference genomes, transcriptomes, metabolomes, respectively, we will not be able to get a high-quality evaluation of the data. And the analysis of environmental samples needs to go hand in hand with lab-based reference samples. And this is what we're doing in our group. And with that, at the end of my presentation, I would like to acknowledge all the people who have contributed to the data presented in this talk, particularly Jan and Alex, who were my PhD supervisors, because most of the data presented here was part of my PhD. Would like to thank the Helmholtz Association as funding my postdoc position, and Leeds, who funded my PhD, and then some additional funding sources that supported my fieldwork during my PhD. And of course, a big thank you to you for listening. And I'm happy to take questions if there are any, either now or also later by email or Twitter. You can see my contact details on the top of the slide. Thank you.
All right, thank you very much, Steffi. That was very intriguing. I have a question right off the bat that relates immediately to obtaining these reference genomes. So can you maybe explain a little more detail about the process of culturing some of these needed organisms, or the way that you're going about that?
So we are not doing any culturing in our lab, but we're working with people who do the culturing, and we also tried it. But the problem is, I mean, they're most happy in snow and ice and not liquid water. Yeah. It's also already something very difficult to simulate in the lab. Yeah. Like snow or ice, and then in sterile conditions. Yeah. Yeah. Then also to have like the right basically nutrient cocktail, because they only need a little bit and not too much. Uh-huh. So I might have missed this. Are you also looking at single cell genomics, or is that not doable for these algae? Not yet, but that's definitely something we are planning on doing in the future. Yeah. Well, that's because, really... I mean, because they're so large, I think only slowly single cell genomics is actually able to do this on these big genomes. Yeah. That's really challenging for sure. Yeah. Do we have any questions within the Hangout? You're welcome to unmute yourself and pop in and ask Steffi a question directly. If you're listening elsewhere, please tweet questions to microseminar, the hashtag #MuSeminar, if you have any at the moment.
Hi, Steffi. I have a question. I'm Lizzie Wilbanks. Nice to meet you.
Nice to meet you.
I was interested in sort of thinking about the role of the bacteria in supporting your ecosystem and what you think they might be provisioning or taking from sort of the algae as the primary producers.
Yeah, so I definitely think they're taking a lot, like because the algae doing a lot of photosynthesis and provide carbon for the heterotrophic bacteria. But that's definitely like an interesting question to study, how much actually the algae also take from the bacteria, because we don't know. I mean, it's quite likely that the bacteria are taking the organic carbon from the algae, but we don't really know the other way around.
Cool, thanks.
But it's definitely for the future.
Yeah, that's definitely an intriguing question. Do you think any of your metabolomics data can shed light on that, or is that mostly targeted towards algal compounds, or can you find bacterial compounds or anything in there?
So I think the problem with this is that the algal biomass is so dominant. Mm. So that most signals actually come from the algae.
Okay.
And so what we could do
one of the things that I was
sorry, go ahead. Is basically try to separate algae from bacteria and then do metabolomics on the separated samples. But we've tried all kinds of like separation techniques, but they love to stick together. Mm-hmm. And it's not always easy to separate them.
Mm-hmm.
Yeah, what I was going to say, sort of building on what Cameron was asking, was whether I was just wondering if there are any bacteria that could be auxin producers that would be stimulating the growth of your algae.
Mm-hmm. Mm-hmm. We definitely get different bacterial communities in green and red snow, so which is quite interesting. So so the algae have a different metabolism, and then also the bacterial community seems to be different. So there seems to be definitely a link.
Interesting. Are there any other questions? Okay. Well, thank you very much, Steffi. So just to remind everybody, you're at Steffi Lutz
yes.
on Twitter, and then I guess you had a slide with your contact information if people want to ask you later.
Yeah, it's on the first and the last slide soon.
Perfect. Okay. Well, thanks very much for your presentation. This was wonderful.
Yeah. Thank you.
Okay.
Mm-hmm.